Okay, take a look at this vintage public health poster for a second. Careless spitting, coughing, sneezing spread influenza and tuberculosis. It's pretty stark, it's direct, and it's a powerful reminder of exactly how long humanity has been battling this invisible enemy. Mycobacterium tuberculosis isn't a new threat. It is an ancient, highly persistent pathogen that has been shadowing humanity for centuries. But despite how long we've known about it, this microscopic organism just continues to outsmart our immune systems and our medicine.
Okay, let's dive right into this. Welcome to today's Explainer, where we are going to unpack the science, the scale, and the sheer survival instinct of tuberculosis. We're literally going to look at the biology of how this pathogen turns our own bodies into a cellular prison, and how clinicians have to play detective just to track it down.
Here is our roadmap for today. 1. A global health crisis. 2. The waxy armor of TB. 3. Airborne invasion and pathology. 4. Spotting the symptoms. 5. The detective work, diagnosis. And finally, 6. Prevention and the future.
Section 1. A global health crisis. Now, to truly appreciate the biology here, we first got to understand the sheer scale of this pathogen's impact.
The numbers are just staggering. About a quarter of the entire global population is estimated to have been infected with TB bacteria. Think about that for a second. 1 in 4 people. Now, thankfully, only about 5 to 10% of those infected will eventually develop symptoms and active TB disease, but the burden is super heavily concentrated. Looking at the incidence rates, you see these really intense concentrations across Africa and Asia. Countries like India, Indonesia, China, and the Democratic Republic of the Congo bear a massive portion of this global weight. And why does this matter? Well, because in a highly interconnected world, an airborne pathogen of this magnitude impacts global health security for absolutely all of us.
To put a finer point on this, let's zoom in on Kenya, which is classified as a high-burden country for TB, TB/HIV co-infection, and drug-resistant TB. In 2024 alone, Kenya recorded 124,000 cases. It is literally the 4th leading cause of death in the entire country. And the demographics tell a really crucial, albeit heartbreaking story. Men, specifically adults aged 25 to 44, are the worst affected. Furthermore, the high prevalence of HIV significantly contributes to this burden, as individuals with compromised immune systems are vastly more susceptible to TB. It's this devastating intersection of vulnerabilities that demands our attention.
Section 2. The waxy armor of TB. So how does this bacteria survive so well? The answer lies in its unique biological profile.
The absolute secret weapon of Mycobacterium tuberculosis is something called mycolic acids. These are basically complex lipids that make up over 50% of the dry weight of the bacteria's cell wall. This isn't just some standard protective layer. It's an extreme, waxy armor that's totally responsible for the pathogen's virulence and its crazy ability to resist being destroyed by our standard biological defenses. And this brilliantly illustrates what I mean. Because compared to your standard gram-positive or gram-negative bacteria, the mycobacterial cell wall is basically a towering, lipid-rich fortress. It is just loaded with lipoarabomannan and mycolates. And because this wall is so incredibly dense and waxy, the bacteria actually multiplies really slowly, taking 18 to 24 hours just for one single generation. But here's the thing, this slow growth is actually part of its brilliant, insidious strategy. It allows the bacteria to remain a dormant, non-spore-forming sleeper agent in the body for years, perfectly protected by that armor.
Section 3. Airborne invasion and pathology. Okay, so how does this armored invader actually get inside us?
Well, TB is a strictly airborne threat. When an infectious person coughs, sneezes, or frankly even speaks, they expel these tiny aerosolized droplet nuclei. We are talking about up to 3,000 infectious nuclei from a single cough. And these droplets are incredibly small, under 5 to 10 micrometers in size, which means they don't just drop to the floor like heavier particles. No, they can literally remain suspended in the air for several hours, completely invisible, just waiting to be inhaled by a new host. Once they're inhaled, the bacteria bypass the natural defenses of the upper respiratory tract and settle deep down into the lungs. This is where our immune system's first responders, the alveolar macrophages, arrive on the scene rapidly to engulf and destroy the invaders. But remember that waxy armor we just talked about? Yeah. The TB bacteria actually inhibit the acidification process that the macrophage uses to digest pathogens. So instead of being destroyed, the bacteria survive and multiply freely right inside the very immune cell that was sent to kill them.
Eventually, the host macrophage just ruptures, releasing a newly multiplied army of mycobacteria. It is, quite literally, the ultimate Trojan horse maneuver. Now, what's really interesting about this slide is how the body responds to that internal breach. Realizing it can't easily kill the bacteria, the immune system decides to just wall it off. Macrophages, lymphocytes, and fibroblasts rush to the site and build this cellular prison around the bacteria. This structure is called a granuloma, or a Gans focus, and while it successfully traps the multiplying bacteria and stops the immediate spread, it also creates an environment where the bacilli can just lie dormant for years, basically biding their time, waiting for the host's immune system to weaken so they can reactivate.
Section 4. Spotting the symptoms. Naturally, this intense microscopic battle eventually takes a real physical toll on the patient.
When the disease is active in the lungs, pulmonary TB presents with a chronic productive cough, fever, extreme weight loss, and some terrifying systemic signs like hemoptysis, which is coughing up blood, and drenching night sweats. But the bacteria don't always just stay in the lungs. Through the bloodstream, they can actually escape and seed themselves in other organs, resulting in extrapulmonary TB. And the versatility of this pathogen here is deeply alarming. I mean, it can manifest as painless swelling in the neck lymph nodes, known as tuberculous lymphadenitis. It could ruthlessly attack the central nervous system as TB meningitis, or it can even invade the genital urinary tract.
Section 5. The detective work. Diagnosis. Because it is so elusive, tracking down this pathogen requires some serious clinical investigation.
Clinicians have a few tools at their disposal here. A chest X-ray is rapid and lets doctors spot abnormalities. Specifically, they're looking for infiltrates or cavitary lesions that are typically found in the upper lungs during active TB. However, an X-ray lacks specificity. So to actually check for an immune response to the bacteria, doctors use the Mantoux tuberculin skin test. They inject a tiny amount of purified protein derivative just under the skin. Then, 48 to 72 hours later, they measure the localized swelling, or induration. A swelling of greater than 10 millimeters generally confirms the person is a tuberculin reactor, meaning their immune system has, at some point, been primed by the bacteria.
But to truly confirm the culprit, the lab has to actually see it under a microscope. And because of that waxy, lipid-rich cell wall we mentioned, standard bacterial stains, they simply bounce right off. Instead, microbiologists rely on a really clever trick called the Zeal-Nielsen stain. First, they apply a primary stain called carbol-fusin. This is lipid-soluble and uses heat to deeply penetrate that waxy armor. Next, they hit it with a harsh acid-alcohol decolorizer. Normal cells will completely lose their color, but the TB bacteria are what we call acid-fast. They stubbornly hold on to that red dye. Finally, a counterstain like methylene blue is applied to color the background.
So, the crucial point is, after this brilliant chemical process, the acid-fast bacilli appear as these distinct, bright red, rod-shaped bacteria standing out sharply against the blue background. This visual confirmation is incredibly satisfying from a diagnostic perspective, because the elusive armored pathogen is finally unmasked and caught red-handed.
Section 6. Prevention and the future. Knowing how it works and how to find it is really only half the battle.
Stopping transmission requires a totally multi-front defense. At the clinical level, the absolute best way to stop the spread is to isolate infectious persons and provide effective treatment as soon as humanly possible. But you have to understand TB is just as much a social disease as a medical one. Mitigating risk factors is essential. We have to address socioeconomic status, crowded congregate settings like prisons or shelters, and co-infections like HIV, which, as we saw earlier, severely weaken the host's immune system. And then, of course, there is vaccination.
Currently, the live attenuated BCG vaccine is our only available vaccine. And look, while it is a critical tool that effectively prevents severe forms of pulmonary TB disease in infants and young children, it has some pretty serious limitations. Chiefly, it offers highly inconsistent efficacy against pulmonary disease in adolescents and young adults. It is quite literally a century-old shield in a modern war, and unfortunately, it really struggles to protect the older populations who are most actively spreading the disease today.
Which leaves us looking toward the horizon. Mycobacterium tuberculosis is an absolute master of survival, utilizing its waxy armor and Trojan horse tactics to persist inside humanity for millennia. With TB still claiming well over 1 million lives every single year globally, the arms race between human science and bacterial evolution is far from over. Will the development of a new, fully effective adult vaccine finally be the key to breaking that waxy armor once and for all? It's the critical question driving researchers today. Thanks so much for joining me on this explainer. Keep questioning, keep learning, and I'll catch you next time.